Hello everybody. Today, our webinar is broadcasted from the chemical laboratory. You can see it at the background. Well, let's begin as everybody is here. Let's start. Our webinar today is attended by myself and the head of our research team. I'm going to introduce him to you later, but now some basic information for you, and then let's switch to the research and scientific part. So the first question that has been actively debated recently is the dividend accruals on the balsam profit. The decision is not yet taken. Some are in favor of this, some are not. I remember that from the very beginning, the idea was that the entire profit is to be divided for the entire company's shares, and given all those things, the decision is not taken yet. Everything remains as it is. That goes without saying that we want everybody to be happy, and this decision is not a big deal for the company. This is mainly impacting investors' profit because the company doesn't care much about how many dividends or shares we have to divide our profit. This is the first point. The second point that I want to raise at the moment is that I have already told you at the previous webinar that our t-shirts are coming. They arrived from St. Petersburg yesterday, and today they are being printed. Today and tomorrow the trial run for printing will take place, and Friday, Saturday the t-shirts would be available to us and would be sent to those who have ordered them. As for the size, we are going to measure all t-shirts and would post information on the eShop for XL or L size, width, length, so for you to define what kind of size you need. Please put a plus sign on the chat if you hear me well, loud and clear. Thank you. Thank you. Next, once again, if you are not aware of it, our personal accounts, your personal accounts have New Year and Christmas options, including balsam, our branded t-shirts, and it depends on which package you choose. And on top, there is an extra discount for company shares. This is our main news. And now let's switch to the main part of our webinar today. Our preparation that we have been working on. As I said a few days in the chat, a few melanin samples have been transferred to in vitro tests. Later about this in more detail, but now please be reminded about the mission and goal of our company. When we started, our primary goal was to create a preparation and to start development of a drug, medicinal drug against cancer, to be affordable for the people and to be safe for human body, because most of cancer drugs have lots of after effects on the body. This is related to the fact that they are killing cancer cells. Those drugs are toxic, and it poisons not only cancer cells but also the entire body of a human being. Unlike other preparations, our drug is based on AstroMelanin. There were a lot of publications about it, and it is not toxic. It has no after effects on other systems of your body, and people of any gender can use it, or of any age, as they have as this medicinal drug has no bad effect on your body. Back in 2000, AstroMelanin was supported by 3 cancer institutes in Russia, and I can show you a document about it. Those who is watching me now can take a look. Heads of 3 Russian cancer centers, Trapeznikov, Parishnikov, and Sipos, they wrote a letter to support that preparation to the Russian president as for the development. At the moment, I am giving the floor to our senior research fellow who is in charge of the development process and everything related to research and development. His name is Stanislav. All right? The floor is yours. He's going to tell you about the processes that we have already passed and what we are doing now. To start with, I would like to make a few comments about the developments based on Mrs. Lach, that you know about AstroMelanin. What corrections have to be made? You need to know that the letter that you can see and recommendations have been written during its development. It was not possible to speak about the full clinical picture at that moment, and it's not possible to speak about it now too, because at that time it was not just possible to find the polymer, which is the active substance. Second, another point to be taken into account. Actually, there is no full understanding on the action mechanism of melanin, and there is no direct relation to treating cancer diseases. Question: do you mean that the mechanics are not clear, right? Right. And the action itself on the cancer cell is very specific. There are many types of cancer, and we cannot say about universal nature of it for you to know that this is not a magic pill. Moreover, now we are talking about a food supplement. A food supplement is a preparation that can act as a supportive therapy, because we have never done preclinical trials on a larger scale. We are going to do it now. Neither for dosage nor for the administration method of this preparation. For instance, orally, intravenous, sublingual. There is no understanding about that. But there is one thing that can be considered as our know-how, which is the point that it was stated indirectly that the best way of this substance administration is a solid way in capsules. What is the advantage? The advantage is in accurate dosing. Leach was using water solution, and it was hard to imagine that every certain volume of water has the same concentration of the active substance that was necessary to give some therapeutic assistance or effect. There were just a few patterns information for you to understand before I get to the main part. And there is another point worth mentioning that we have been working on, and you may have a question. If everything has been done, why not replicating it? Actually, not much has been done in terms of clinical stage. First of all, this is related to the fact that the dosage had to be specified. There are many publications by Mrs. Leach, and in patents, different dosage was specified. What is the correlation between the type? And it was just Leach's assumption. Like, this is good for immunity, so it's like a classical herbal nutrient or supplement that would kill cell necrosis. In plain English, this is removing a negative effect of necrosis, because if cells are dying, you have apoptosis, and we don't have a lot of free radicals at it that have no big impact on the human body, but necrosis of blood cells that are happening under certain impact, as well as metabolism, which is disturbed, and any other impacts and bad habits. In this case, yes, your body may get huge doses of free radicals, and their direct adverse effect is not proven yet, how they are related to tumor processes. And you need to know what. Actually, the so-called cancer cells are producing unnecessary proteins, and they are produced hundreds of thousands during our life, and the immune system detects them and kills them during phagocytosis. But at a certain moment, that process is disturbed. If someone tells you when it happens, this is not possible to know, and no one can tell you why and when. So this idea that that you need to improve your immune system, that would affect those cells and would also be in a good concentration, which are T killers during phagocytosis. This is just a common phrase. Generally speaking, it is right, but actually it is wrong, because immune system and is different from another person's immune system, because now you need to support certain parts of the immune system. Sometimes it's bad, and it could result in cytokine storm, and in certain ways. And it was considered by Leach herself that the impact of melanin and its antioxidant activities was cleaning blood and lymphatic system from adverse products of cellular decomposition and life. Like, we are made of what we eat, what we breathe, and so on. It has some ground, and in this respect, this AstroMelanin, the food supplement that was promoted by Liach, this was a good part of the story, a true part of the story, because you could touch the stuff. We had in vivo tests supporting those ideas, but you need to know what the statistics and selection of those researches is low, and it's hard to say definitely how universal it was. This is the first disadvantage of the research that have been conducted up until today. The second disadvantage is the way of delivery of that drug in the human system. Those tests were taken by an American institute, I forgot the name, but there is an article about that, a research team leader feedback, and they were giving quite a good feedback about melanin and its action on a cellular or on a cells level. But they were saying that it takes quite serious research in terms of its delivery into a human body as a solution per oral administration, or like candles or suppositories. It was not resolved, and it's not clear why they began or continued to dissolve melanin in water, and that was considered that the concentration in every volume was the same, and that was considered to have a therapeutic impact. But that is very disputable, and I'm going to tell you why. This is the second point of the second weakest point in the research that has been conducted so far, the dosage and administration method. And the third point is melanin composition. Melanin is different. There are animal and herbal types of melanin. They are different. They differ a lot by properties, and it's not clear why, because to define a clear structure of melanin or melanin-like systems that are part of that common polymer, it has not been possible to do by anyone. There is synthetic melanin when you take elementary cell and you try to connect them or to sew a big polymer that can be considered melanin as well, because it has certain structures and supplements and such. But this is synthetic melanin, and if we do it in the same way as we did the natural or native melanin, their properties differ, and it doesn't have such a bright or robust impact as the natural melanin has. And the reason is not clear. Why am I telling you this is for you to know what is the task that we are facing. We want to resolve all those issues that have been unresolved so far, or they were tackled with strange ways, and it's not possible to clarify it as a scientific point. So we were trying not just to resolve it, but to reproduce certain results that would allow us to say, yes, we can be clear that this thing has therapeutic action. Here it has no therapeutic action, or in certain ways you may say that it could be effective in line with other medications for this or that disease. My question: where was equipment available when Lech was alive to do the same as we do today? Answer: what was available at that time, the same effect was possible to do as we have. This is EPR, that researches into antioxidant activities. You probably heard such data in previous versions, like we took EPR and we get 10 to the degree 19 spins per gram. What does it mean? This is a stable, this is a well-established measurements of EPR, and they are directly correlated with antioxidant activities. The higher this metric, the more effective is the antioxidant activities. You need to know that antioxidant impact is not directly related to anti-cancer performance. This is a myth. Like, many people think that tomatoes have increased antioxidant activities, but I've never heard that cancer drugs were treated with tomatoes. But why melanin is special? Because Lech was working with animal melanin, non-vegetable or just vegetation melanins. But for animal melanins, it was proven that they had antioxidant action related to anti-cancer activities or performance. And having read that database and literature, and due to the fact that synthetic melanin was also available, there was a question: what is the composition of the native melanin? Frankly speaking, at the moment, we don't know. Well, we can judge, or we can guess, but as far as the melanin composition is concerned, we have been testing a lot related to the EMR spectroscopy that gives us certain answers. Why do we need it? We need it to have a chance to speak about the active substance, and this is the major problem and the major difference between a herbal supplement or dietary supplement and a medicinal drug. If we specify what is the active substance, we can say that this is a medicinal drug. This is for your understanding. Now, what we're able for us to do at the moment, we were able to cultivate a strain on a petri dish, the strain that was used by Lech, but during the test it was discovered that some strains are simpler to work with, and we can cultivate more strains per volume, so we can cultivate more of that substance per a unit of volume of those fungi with melanin, rather than growing microfungi. And our task now is to show if there is a difference in vitro tests at cell level, or if there is no difference. This is the first point. And secondly, the same difference has to be proven on mice in vivo. So this is the first part that we decided to do. So we took two strains in parallel, and we were trying to get melanin from them. But I think that this is the same melanin. The major problem was to get melanin from the strain culture. All the time we were trying to select a generation methodology because the data that we had in patents and in publications, they were not replicated. They were either written like this to replicate easily, or the description was not very clear, so they were poorly replicated. And we were able to select our own replication method. Then came an issue of melanin generation from the fungi cultivation. The problem was also resolved, and we have a clear understanding of maximum generation. But then came another point. You probably heard the water-soluble and non-soluble melanins, but this is basically the same melanin, but melanin as a polymer has functional groups that can be replaced, and that melanin could become water-soluble. But in practice, it turned out that to reach full water solubility is not easy. There were methods of melanin, this polymer generation, where we can give a solution of water alcohol and water solutions. Why is it important? This is probably not important for the final pharmaceutical substance that can be taken into the capsule. First of all, this is important for testing because they work with water-soluble forms, and to prove that our polymers are effective, we need to cite this formula. Well, you're going too deep. People are about to fall asleep. Well, if you have any questions, please post it in the chat. Look, we have given it for in vitro tests a few types of melanin, a few types, I mean, from different strains. This is the first point. And second, different forms, different generated forms, which is also important to know what is a working substance. At the moment, we have 4 samples. Well, partially, we would get data in late December, but the full research would be available by the end of next January. Question: in vitro is cell tests, right? These are tests on cells, and we are trying to understand whether there is a link between antioxidant activities and cytotoxicity. If we are not talking about the signal proteins, this is another therapy. Speaking about chemotherapy as a classic way of treating cancer, cytotoxicity is the major characteristic telling us about the effectiveness of decomposition of cancer cells. As said earlier, other cells are also damaged, and the targeting way of impact is the problem for the entire anti-cancer drug industry. Question: how much time would last in vitro tests? Answer: 2 weeks in December, as we hope, and another 2-3 weeks in January. So I think that late January, early February, we could have some data. All right, let me look through the questions. Well, the question is, can we administer melanin by HIV/AIDS infected people? Answer: there were no contradictions against the drug. It showed no toxicity at toxicity tests, so there are no contradictions. But as for the positive effect for HIV/AIDS people, still no data, so it's hard to say. You will never get poisoned, but to whether it's going to be whether you're going to be cured, we have no data yet. Question: can we sell melanin for $100,000 or $1 million? There are many drugs that sell well, that are made of. I cannot read this question. It's not clear. Speaking about the sales volume, I have said about it at the last webinar. Well, of course, we will not be able to sell a lot for billions of dollars in the first or the second year, because to start selling like this, we need a huge network all over the world, a well-established network. If we take major pharmaceutical companies, well, they have spent dozens of years to establish sales markets, and to boost sales volumes, we need to partner with a major company and to work together. This is the answer for your question. If we do that on our own, provided that our drug would have an effect that we want to have, and that were supported by Liach before, this is realistic, but it takes time. It's not a year or two. You need to know that. Right. Any medicinal drug development, even not against cancer, but it takes 10 to 15 years, given the third clinical test phase. In our case, this is a herbal nutrient or dietary supplement. When we confirm that it's active, we can start using it much faster, because the medicinal drugs have more complicated tests, and this is not required for dietary supplements. Well, once we have in vitro test results, we would be able to register it as a dietary supplement and start selling it. Right. And in parallel, we could just provide cytotoxicity, chronic toxicity, impact on cardiovascular system, on tissues. So it's pretty easy and quick to do. It takes 3 to 4 months. And if all results are satisfactory, and we clearly see that it's not going to be harmful, that could be used as a dietary supplement, because if there are confirmed data on in vitro and in vivo tests, that could be done. The only thing that I want to specify, you need to know that we cannot say or speak about universal nature of that drug. That could be a recommendation. That would be not possible to answer, because any cancer has some common points, but every cancer is different. And as we're trying to understand the mechanics, how it works, before that moment, we could only be limited by words that, all right, for special in vitro or in vivo tests, that could be done on many types. That can work out, and that would be the most honest thing that we could say. Question: what about the patent protection of this preparation? Well, we're writing a patent. We're lacking some composition data as well as in vivo tests results, but as for generation, the patent, or not a patent, but it's a boilerplate is there, and once we get more data, we would apply for a patent. Question: what about in vitro tests? Will it show where to go? Well, it will. It's going to show whether it's going to be used as a therapeutical active composition or not. That's the in vivo tests would show. Moreover, we're going to use it as a dietary supplement in capsules, not solutions or crystals, not dissolution of certain crystals in a certain amount of water, as it was the case, because a clear dosage for a person to know that he administered as much of it, because colloid story, you would never see it with your own eyes. There is no full dissolution, given the fact that you dissolve as much melanin per 1 liter of water. You will never see it with your own eye, but a capsule would allow you to have a certain dosage, and then we could watch infection. Question: is there any understanding on all AstroMelanin costs and time frame? Will it be a dietary supplement or a medicinal drug? That's already answered. But as for the costs and the timeline, we have information. Now we are in the process of drafting slides. Now the timeline is the following, based on in vivo and in vitro tests. In vitro tests early February, and in vivo, how much will it take? Well, in vivo tests, I think, would be made or done within 3 months' time. The problem is that we're trying to find certain types of mice that can produce certain types of cancer, and only after that we can do it. I think we can do it within January and 2 months for tests, so it's late March. Yeah. What about the Rusich balsam sales? Right. I was talking at the previous webinar that we were upgrading our eShop, our documents, etiquette and packaging and marketing materials to boost sales. Our marketing people have been working on it now. What is the bottom line? Do we want to maximize development for AstroMelanin or what? Answer: yes, this is an option. What is the price, the minimum price for development? Answer: We have already provided answer for that question. As for the development costs, we remember that initially we began working with the FRAN preparation. So the research was done into its development, so we can be based on those documents. So the development costs were about $3 billion. It was 10 years ago, but given inflation, the minimum figure is like this. How long will the marketing team prepare or work out? What do you mean? This is a brand new product in the market, and to market a new product, to boost sales, it's not possible to do. You can take many products that are known today, and once they appeared in the sales, they were not in demand. So marketing is a topic that you need to go in, and you need to test a few options, and when you find a workable option, you can use it further. So to say that there are no results after 1 year, it's not quite true. The results are there, but this is a research result. We need to know, we already know what does work, what doesn't, and what has to be done. As for the marketing team, we have changed it for the third time, and it's a problem to find a good team of marketing people who knows what to do correctly. So that's another line of activities. What is stronger, melanin or FRAN? That's a question about nothing, because they were not compared. To get a comparison, you need to have clinical trial tests available for FRAN and AstroMelanin. In this case, you need to have a base. You need to compare apples and apples. If you compare apples and oranges, you can only just judge by size or availability of skin or number of kernels. So it's general tests that are, averaged on a certain basis. Only those tests with a common denominator could be truthful. Nothing to comment on this now. If you're going to sell AstroMelanin to Pfizer, will you sell and pay dividends, or will you pay one-off and that's it? Answer: if we sell the preparation to Pfizer, the profit will be distributed by the number of shares, that's clear. And for research sales, no one is planning to go away, because the sales are ongoing, production is ongoing, and we want to continue it. And there is an idea that even if we sell off AstroMelanin, we would be able to produce it as a herbal nutrient, and the sales would be closed in our communities. It will not be available in shops or anywhere else, but only for company partners. Question: what do laboratory personnel think about Rusich balsam? What do you think about Rusich balsam? Can you explain it to me? Is this the balsam that you gave to me? Yeah, it's Rusich, exactly. I didn't know that it was named Rusich. Well, what I can tell you is that This is a dietary supplement with no clinical trials conducted on it. Similar to AstroMelanin, I can tell you nothing. The only thing is that I tried it myself. I gave it to my children and my parents, and during the time of flu peaks, I'm positive about it personally, because I have noticed the effect. I have not noticed about my children, but I have noticed it a lot on my parents, because they have a few chronic diseases, and the effectiveness of this constant. Once you have a drought, they fall ill, and that is now less. Can it be considered an exception to the rule? I have no statistics about that, but I admit it myself. In winter, spring, or autumn, winter. I'm not trying to convince you, but there is certain sense behind it. But to speak about a scientific base that I could just put under it, the answer is it's a popular science, I would say, rather than a scientific thing. Any other questions? Or should we call it a webinar? Well, there are no more questions. Well, if there are no more questions and everybody knows everything, I wish you a good night, and bye-bye, and until next time.